Continuing on with one more set of math notes. Sometimes in biology experiments, we need to use percentages to be able to compare different things to each other. And so one method we could use is called percent change. Percent change assumes that there is an original or old value that you're comparing a newer value to. And so it will use the old or initial value as the reference value. And ultimately what we're trying to do is see if our new value is the same or if it has increased or decreased compared to the initial or original value. So again, we do have a reference value, and the reference value is the old or initial or original value. Here's an example. If we had a plant that grew to 60 centimeters tall from 55, so within that time period, it grew 5 centimeters, and we wanted to calculate the percent change of the plant's height over that time period. We would use 55 as the old or initial height, and then we would use 60 as the new height. When we put that into the formula and multiply it by 100, so it can be expressed as a percentage, we would be able to say that the plant experienced 9% positive growth during that time. Notice that this isn't absolute value, and so it's very possible that we could get a negative percent at the end of this. If you were looking at something that got smaller in size, right, then the new value would be smaller than the old value, and so our final answer would be negative. That would mean it decreased in size. So it does matter that you actually do put the final value here and then the initial value. Sometimes I have students want to swap these and just put the bigger number first, but it isn't about that. We are looking for, did it stay the same, increase, or decrease relative to the original. So again, it is possible your final answer could be negative. We could also look at what's called percent difference. In percent difference, we don't really have a reference value to compare to because neither of the two values that we are using to calculate this has any reason to indicate one over the other. Like we don't have a new and an old value. It's like you're looking at two totally different values and then comparing those in a percent to see how different they are. So our reference value has to be calculated as the average between those two values. So here is an example for this. If we have two sticks, right, and one we've measured to be 100 centimeters long and the other we've measured to be 50 centimeters long, well, there's really no connection between those two sticks necessarily. We're just trying to see what is the difference in size between them in percentage. And so we can calculate percent differences. But again, it's not like we have an old or a new value because they're two different sticks. And so our denominator this time is going to be the average between those two values, whereas the numerator this time is the absolute value of the difference between the two. Because again, we're not looking at growth or anything like that. It's two different objects. And so since it's absolute value, it really wouldn't matter which one you put first in this case. So in our example, 100 minus 50, and it would be the absolute value of that. So even if you did 50 minus 100, it would still be a positive number. And then divided by the average of those two values, so 150 divided by 2. And then we're going to take that number and multiply it by 100 so it can be a percentage. So 66.67%. That's the percent difference between the lengths of our two sticks. R versus R squared value. These will become important in your IA. Almost all the time in an IA, you will talk about these two. Notice that R is always lowercase, but in R squared, the R is uppercase. They are related terms, but they also are used to determine different things. R is the correlation coefficients. What it's really looking for is when you graph two variables on a graph, is there a linear relationship between those two variables? And so that's what it's looking for. And if there is a linear relationship, then is it positive or negative, and how strongly correlated are those two variables to each other? The R value can be positive or it can be negative. So it's going to be a number between 0 and 1. But depending on the trend of the line, if it's showing a positive slope, if the line is increasing, then it would be a positive number between 0 and 1. But if the slope is negative, if the line is decreasing, then it's going to be a negative number between 0 and negative 1. And then the farther away you go from 0 in either direction will be less of an indication of a linear relationship. So we want it to be close to 1 or negative 1. We don't want the number to be like close to 0 in either direction. R squared stands for the coefficient of determination. In R squared, we're not looking for a positive or negative anything. In fact, R squared will always be positive because the value is squared. What it's looking for is how close do the data points fall to like the trend line. Like how close is the data together? What is the tightness between the relationship of the two variables? And it usually is used when we are implying that there is some sort of causation between the two variables, like one caused the other or affected the other. Typically in your lab report, you will talk about R squared first because it will be calculated using sheets or Excel like by inserting a trend line, and then you'll need that value to be able to calculate R. So it just makes sense that you'll talk about this one first, and that will roll you into R value discussions in your IA. So again, we are looking for, is it a good fit to the model? Like, do these points fall close to the line? The higher the R squared value, the better. R squared varies from 0 to 1, and again, it's always going to be positive. If R was equal to 1, then every point would be on the line. So again, we want the number to be closer to 1, not closer to 0. The higher the R squared value, the better. Outliers will bring down this R squared value. So on this example, we've graphed budding day of the year and an average spring temperature. And so we inserted a trend line on our scatter plot. The equation is there. And then of this trend line, we also displayed the R squared value, which was 0.499. So then we would be able to say something like this. On the data shown below, we can say 49.9% of the variation in budding day can be explained by our model relating budding day to average spring temperature. And again, ideally, we would want high R squared values. We talked previously about when R squared is low, then you may have to process the data with and without outliers to see, is it ultimately going to make a difference in your statistical test result. R is going to be looking more for patterns in things. Remember, we are looking for, is there a correlation between whatever you've graphed on your X versus Y axis? Between your two variables, is there a correlation? And if there is, how strongly correlated are your two variables? And is the relationship positive or negative? And so R, remember, varies from -1 to +1. And the farther you go from 0 in either direction, the better it is, right? So the more strongly correlated something is. So we want like values close to -1 or values close to +1. R would be calculated from the R squared. So you've already displayed the R squared value on your graph. And and then you'll just take the square root of it in order to calculate the R value. If this was in your IA, you would have to explain how you got your R value. So you would just explain it kind of as it's stated on this slide. Notice that in this picture right here, our line is increasing, our slope is positive, and so we would be able to conclude that there is a positive correlation between X and Y, versus on this graph right here, you can see that the slope is decreasing, so there is a negative correlation. So in this one, as one increases, the other increases, but in this one, as one increases, the other is decreasing. It can be assumed that since you are calculating R from R squared values, that high R squared values will lead you to high R values. So again, we're going to be discussing these together because one is calculated from the other. You will need to talk about both in the conclusion section of your IA. And then again, the higher the R values are, you're also commenting on the strength of the correlation. So high R squared value or high R values would mean not only, you could say that there's a strong positive correlation or a weak positive correlation, and the same for the negative. The last thing we'll talk about is serial dilutions. Oftentimes you will make a serial dilution if you need to dilute something further down to a much lower concentration to use for something else in your experiment. And they are typically made by making the same dilution step over and over and over and over again, where you use the previous solution to go into the next solution. Every step is going to be the same. So the dilution fold is going to be the same in every single step. This is called a geometric series because it means we are maintaining a constant ratio between adjacent solutions. And so here we have a stock solution. So this is whatever we're initially starting with. The volume in this solution does not actually matter, but how much you take out and put into the next solution does. So in this case, we're doing what's called a 1 in 10 serial dilution. That means we're going to take one part out. So whatever number this is, it wouldn't matter. In this case, it's 100 microliters. That's going to count as our one part. We're going to put it into a different container. But that is just one part of the 10. So the other 9 parts would be water, and we just diluted it. And then we're going to do it again. So we're going to take it out of this one this time. Same amount this time, 100 microliters. That's going to go into a different tube. That's going to be one part of the 10. And so the other 9 parts to make the 10 is water. And then again, do it again, repeat, right? So 100 microliters from this tube into this tube, that's one of the 10 parts. Add the other 9 parts to get to 10 of water. So that means we're adding 900 microliters of water. And then we just keep doing it. And you can see how much diluted it is every single time. These numbers compare back to the original. Each one is a 1 in 10. But if you look at this one, because we've done 1, 2, 3 dilutions now, this is a 1 in 1,000 dilution compared to the original. By the time we go through all our dilutions, so 1, 2, 3, 4, 5, 6 serial dilutions, this one is a 1 in a million dilution compared to this one. When would we use this? Well, if you wanted to make different concentrations where they increased greatly, right, by a geometric series between each concentration, so maybe in an osmosis experiment or in a plant hormones experiment, you could do it that way, or in an experiment where you might be needing to count bacterial colonies from a culture, but originally there's just way too much bacteria to count. So if we were to try to count this, well, we couldn't see it, right? You can't even see individual colonies because there's just so much. So then we would do a serial dilution, still too much. Do another one, still too much. And you would keep doing that until you can make it countable. So 2 colonies, right? But if we wanted to then estimate how much was present in the original container, then we would just go backwards with the math based on those 2 colonies. And that way we can say we counted 2 colonies and then use that as an estimation for what would have been present in the original container. And it's not always a 1 in 10. This keeps showing a 1 in 10, but it could be a 1 in 5, a 1 in 6, a 1 in 3. It depends on the experiment. But that would still mean the same thing. If it was 1 in 3, it'd be 1 parts, whatever you're transferring from the previous container, and then the other 2 parts to get to 3 would be water, for example. And FYI, going back a slide really quick, I replaced this picture. There used to be a different picture where the picture itself did not quite make sense. It just wasn't drawn correctly from wherever I got it out there in internet land. And then that caused me to change these numbers as well. So you might update that on your note taker. And here's just one more example. So now we're looking at a 1 in 5. Okay, and it tells us to perform 3 1 in 5 dilutions. We're going to start with a stock solution that has a concentration of 5 molar. So if we start with a concentration stock solution of 5 molar, and we take 1 part out and we put it in the next one, and then we add water to get to 5, we've now diluted it. And to figure out what the concentration of this one is, we would divide the original number by 5. Okay, so after 1 dilution, so 1 part of the original one, 4 parts water, we've now made a concentration that's 1 molar. If we do another 1 in 5, same thing, right? So we've taken 1 part out from here, put it into a different container, added 4 parts water, but to figure out what concentration it is, I would divide this by 5, so 0.2 molar. Do it again, divide that by 5, and we now have a 0.04 molar solution concentration. So after 3 1 in 5 dilutions, starting with a 5 molar concentration solution, we've now made a 0.04 molar concentration solution because it is a geometric series. That would make it a 1 in 25 compared to the original stock solution. And that's it. Please go ahead and send me an email or ask me questions if you have any. Oh, here are some extra links in case you need help with some of the science math I'm talking about. So they're just ready to go in the PowerPoint if you need it.